Challis, for this interesting and complex presentation. Any question?
Rob.
Taylor.
John, very nice stuff. Is it known the distribution of the CRH receptors 1 and 2? Is there differential expression at the fetal-maternal interface, and do vascular cells actually have those receptors, or on the decidua? I don't know so much.
Felice is the real expert on that. My understanding is the amniotic epithelial cell expresses CRH1 but not 2. Chorion expresses both 1 and 2. Placental trophoblast both 1 and 2.
In the vasculature, I'm just curious as to whether the vasculature expresses.
In the vasculature, I don't think we know. We do know, of course, from Vicky Clifton's work, that CRH promotes vasoactivity in the placental vascular bed, and perhaps that's one of the, you could make the teleologic argument that one of the responses to an adverse environment, hypoxemia, is to produce CRH, which increases vascularity, blood flow, and increases oxygen delivery and so on.
And that would presumably be CRH receptor 1, right?
Sorry?
That would be receptor 1, would be the presumed pathway there, rather than...
Presumably in that sort of, yes, absolutely.
Yeah, John, could I ask you, so to elaborate a little further on the male predominance in preterm birth in terms of mechanism of action?
Well, we came across it, I just showed you one or two of Mariam Yaganagi's slides, and the data are all published, but marked differences in trophoblast cells in their ability to respond to LPS with prostaglandin synthesis production being higher in trophoblast from a pregnancy with a male fetus. Prostaglandin dehydrogenase, if you like, the protective enzyme, it's upregulated in trophoblast cells where there's a female fetus. So it does look as though when you start to put it all together, IL-10 is upregulated by LPS in both tissues, and the GR1 supernatant fraction adds additively with LPS in that effect. So there's about a 1% overall difference between males and females. That's obviously pretty significant in large numbers. Our suggestion from these data at the moment is that it does appear that the male fetus develops in a more, if you will, pro-inflammatory environment and is more susceptible, particularly to the infection-driven part of preterm birth. And that's when, on that the slide from of the series of analyses, the earlier time there, the 30 weeks or so, predominantly probably infection-driven. Our sense is that it's the later preterm births which are more HPA stress, maternal HPA stress-driven births.
Okay.
But a great area for investigation.
Another question?
Yeah.
Congratulations for your nice topic.
Thank you.
I would like to ask you, do you know if there is any experience with the opposite model, the post-mature birth?
I'm sorry, I'm a little deaf here. I think we probably start with Felice. I mean, the CRH concentrations, as Roger Smith showed, in post-mature are much lower and go up later. Felice, do you want to comment on this?
We have also some sex difference like
Yeah.
shown in the
Yeah.
John, I was wondering if some of the sex differences relates to the fact that at any gestational age, maybe males may be in a different maturational state than females in gestations. And we know that surfactant, pulmonary surfactant, is actually expressed later in males and females. And I just wondered if some of the interaction actually had to do with the fact that they're the same gestational age, but they may not be the same exact developmental age.
Yeah, no, so I think, I can gossip about what I think is going on and speculate. I mean, I think it's clear from a number of studies, and again, stimulation of surfactant is one response. I think others have been referring to that today, of the male. Male just lives in, I think, in a more precarious circumstance for much of pregnancy than does the female. And, I think in Vicky Clifton's work, for example, looking at the effects of synthetic glucocorticoids for women managed with them for asthma through the course of gestation, you see exactly the same sorts of sex differences, and again in Vicky's work, some sex differences, again in different cytokine being produced from the placental tissue. So very, very good agreement, I think, with the work that she's done, our work, and the work that has come from Felicia's lab.
Well, thank you again.